molecular basis of heredity is all about connecting what Mendel discovered about traits being passed from parent to offspring to what is the molecule that actually does this. We know that it's DNA because it's already been discovered, but the process of discovering DNA has actually been really important because it has a lot of intricately designed experiments. Now, first we talked about Thomas Hunt Morgan discovering the chromosome. That was nothing big. He just saw it under a microscope. But the fact that he realized that there were linkage patterns, that on a chromosome were many individual genes, that's a really important part that Thomas Hunt Morgan discovered. Remember, he first saw it in sex linkage in his fruit flies, but lots of other genes are linked because a chromosome, remember, is a large portion of DNA. So we're going to have lots of different genes. And so each of these little banding patterns is actually showing you a distinct gene. And so then the next important discovery was a man named Griffith, and he was trying to figure out what actually gives characteristics and the idea of, well, what really is that chromosome? What is it made of? And so he was studying bacteria that cause disease, virulent and non-virulent, no disease, and he discovered that if he damaged what causes the disease, if he heat killed it, they could actually remake that as long as they had what he thought was the DNA. So Griffith is really just trying to prove that it's not the protein that gives the trait. So the fact that we were still virulent after we killed the protein, so heat kill damages the protein, but they can still be virulent. And this is because we know the DNA is just going to make more proteins. So we wanted to prove that DNA was the transforming factor, that it was DNA doing that. So then Avery, McCarty, and McLeod used enzymes. One enzyme broke down the protein. Well, the cell just made more. One enzyme broke down all the RNA. Well, the cell just made more. One enzyme broke down the DNA. The cell can't do anything at this point. The conclusively concluding factor, the way that they 100% proved it, is with an experiment from Hershey and Chase. They actually used a blender. And what they were doing was radioactively tagging. So we might want to add the word tag in there. They used an isotope. Remember, isotopes have different numbers of neutrons, and so they can actually release different wavelengths of light. It's a little more knowledge than we need to know. As much as that we tagged it, they were able to tag the protein and tag the nucleic acid. And what they did is when they put these in a blender, they were actually able to separate the protein, but show that the cell had been infected with the virus. So it was all about showing what actually got inside of the cell. So using viruses and their ability to inject a nucleic acid. Keep in mind, proteins have sulfur, DNA does not. So that's how they were able to tag them differently. So at this point, we have proven that DNA is the transforming factor, or the DNA basically has the code. So now they turn all of their attention to the double helix. Now there is no microscope strong enough to see DNA. Anything that we have of DNA, any pictures, they're drawings or they're sketches or they're animations. There are no pictures really of DNA. What they were able to do is isolate the nucleotides and tell what the nucleotides were made of. This has to do with chromatography, specifically gas chromatography. But the main idea is they were able to use chemistry to figure out what DNA was made of, nucleotides. So Chargaff was actually working with these nucleotides, and he came up with a basic pattern. The pattern that he saw is that the number of purines, which are A and G, pure silver, the number of purines equaled the number of pyrimidines. So A and G equals C and T. Now, he didn't really know what this meant, But it was a fact that no matter which organism you looked at, the amount of A and G equals the amount of C and T. Now we can move these around. We can say A minus C equals G. You can move it all around. And if I give you one, you can figure out everything else. So if I tell you that A is 10, we know now that T is 10. Because what we got later from Watson and Crick is this idea that there are base pairs. So this knowledge right here led to base pairs. When Watson and Crick saw this data, they thought, well, maybe one of these is matched up with one of those. And they were able to figure out that A and T base paired, and G and C base paired. That somehow they were across from each other. Linus Pauling is another scientist who was in this race for the double helix structure, and he studied helices or helixes, and his study of the helix actually really contributed to this. He doesn't get a lot of credit, but his knowledge of the bonding patterns really helped contribute. But the number one thing that led us to this structure, we know Watson and Crick, they get the credit for the structure. But they didn't figure it out all on their own. It was based on all the things before, including Chargaff's data on the number of purines and pyrimidines. And Rosalind Franklin had a big role. She was able to use X-ray crystallography to take pictures of DNA. Photo of DNA. Now the picture that she saw really wasn't very much. All that she could see was an X. Well, they knew DNA wasn't in an X. So they were trying to figure out, well, what is this picture telling us? Like, which way are we looking at the DNA from? And we know, well, they were looking down the middle. And that's why we're seeing that helix idea. But she didn't make this connection. So she's taking thousands of pictures. And Watson and Crick are really trying to figure this out. So they ask if they can see her pictures. She wasn't too fond of that. She had done all this work. They were tinkerers. So they didn't like to be in the lab, but they sure liked to play puzzles. So she was like, you know what, I'll teach you how to take the pictures and you can take your own. They didn't like that. So they convinced someone who worked in her lab, Maurice Wilkins, he's a good enough guy, he shared her pictures. He shared those pictures. And as soon as Watson and Crick saw that, they were like, oh, it's this twisting thing. And they cut out a bunch of pieces, and they played around with it until it fit. They actually made a lot of mistakes along the way. One of them was that they put the sugars and phosphates. Now remember, these are just the nitrogen bases. These four things are just the nitrogen bases. A true nucleotide has a sugar, a phosphate, and a nitrogen base. So they're trying to figure out, how do those three parts fit together? We know now that those three parts fit together with the sugars and the phosphates bonding up and down, and the nitrogen bases hanging on the inside. This is what Watson and Crick were able to tinker around and figure out. So once they assembled these little pieces and they started moving them, they had to use a lot of chemistry to figure out, well, what would bond with what. The first time, they actually tried to put the sugar phosphates on the inside. One interesting thing about a phosphate, if you remember, is a phosphate group is PO4 negative. A phosphate next to another phosphate would never stay. So one thing Rosalind Franklin did is she did kind of try to help them out. She's like, this won't chemically work. Phosphates can't be on the inside, they have to be on the outside. So she contributed a lot, but she doesn't get a lot of credit because she actually passed away from cancer. All those x-rays cause mutations. So by the time Watson, Crick, and Wilkins were awarded the Nobel Prize, Rosalind Franklin had already passed away. So she doesn't really get the credit. So she's sometimes called the Dark Lady because she was adamant, I'm not giving you the answers. Of course, they took it anyway. When we look at the end result of Watson and Crick, which is really amazing, when we look at this end result, we have a lot of information. So our job right now is just to look at how did they get that information, the idea of what is DNA, proving that it was DNA, that we can focus on it, and then the base pairing is important too. So Chargaff's data actually led directly to these base pairs. Okay, so here's what we need to look at. The structure of DNA is double-stranded. What this really means is that there are two polymers. Keep in mind what a polymer is, many parts. The parts of DNA, the parts are nucleotides. And nucleotides have three parts: a sugar, a phosphate, and a nitrogen base. We'll talk about those in just a second. But double-stranded actually means that, imagine if you could cover up one side, that we have one polymer. Sugar, phosphate, sugar, phosphate, sugar, phosphate, nitrogen base is one polymer and another polymer. And they are held together by hydrogen bonds. If you remember these hydrogen bonds, oh goodness, you're going to have to go back to our beginning unit and remember, why is there a hydrogen bond? It's not just because there's a hydrogen there. It's because of polarity. So go back to covalent bonds and polar covalent bonds and the idea of the unequal sharing of electrons. So because these nitrogen bases create a polar moment, that's why they bond together. So when we say it's double-stranded, what we really mean is that there's two separate polymers held together. Well, what is this polymer? The polymer is many nucleotides. Now in DNA, the nucleotides have three specific parts. They have a deoxyribose sugar. They have a phosphate, the PO4 negative. This negative will really come into play when we do DNA technology and engineering. And they have a nitrogen base. Now, there are 4 nitrogen bases: adenine, thymine, guanine, and cytosine. Versus when we get to RNA, we're going to have a different nucleotide. So DNA nucleotides and RNA nucleotides, similar but different. Another unique feature that they had to figure out is actually that when I make this polymer, the sugars and phosphates point in a different direction. We call that anti-parallel. So the sugar and phosphate, we call it the backbone because it's pretty strong. The sugars and phosphates run in opposite directions. And we're going to talk more about this tomorrow. But basically, we're really going to pay attention to the sugar. And we're going to number the sugars. And we're going to be looking for 5' carbon or the 3' carbon. So we're going to see tomorrow that the way these nucleotides orient themselves is either with the 5' up or the 5' down. And you can really see that in this picture, actually. Okay, quick other things. The sugar-phosphate backbone. We're going to have what are called phosphodiester bonds. Phosphodiester bonds. And they are really strong. They're not easily broken. And this is on the outside. The last thing are these complementary base pairs. Super important. Complementary means go together, like complementary colors. So not complement like that's a pretty dress, but that they go together. And we're always going to have a purine bonded to a pyrimidine. And it's going to follow a certain pattern. You should know this: A to T and G to C. But real quick, adenine and thymine have 2 hydrogen bonds. Guanine and cytosine have 3 hydrogen bonds. We get out of school at 2. So if you can remember